We are going to talk about the chemical sensations which include olfaction and gustation. Olfaction is the ability to sense odors through the detection of substances which have been aerosolized into the environment. In common words, it's smell. While gustation is the sensation which is produced by the interaction of taste receptors with solubilized chemical stimuli in the oropharyngeal cavity. In common words, that is taste. Let's start with olfactory system. Olfactory systems start in the roof of the nasal cavity on the inferior surface of the cribriform plate, where is the olfactory mucosa. It is composed of a superficial acellular layer of mucus that covers the olfactory epithelium and underlying lamina propria. The olfactory epithelium contains three main cell types: olfactory receptor neurons, supporting cells, and basal cells. Olfactory receptor neurons are bipolar neurons that has a single thin apical dendrite and a basally located unmyelinated axon. The apical dendrite extends to the surface of the epithelium, where it forms cilia that protrude into the overlying mucus layer. These olfactory cilia contain receptors for odorant molecules. On the cell membrane of these cilia are odorant receptors, which are G-protein coupled receptors that interacts with odor ants and initiate the olfactory signal by generating graded potential or action potential. Humans has almost 1,000 different types of odorant receptors which can bind and recognize 1,000 different odorant. All odorant receptors are second messenger type of receptors. As you know, one of the most important characteristics of the second messenger is signal amplification, which allows detection of lower thresholds of sensations since it is going to be amplified. And that's why olfaction is almost the most potent sensation in our body. Another characteristic of second messenger receptor is no adaptation, hence olfaction sensation has almost no receptor adaptation, and any adaptation is central adaptation. The supporting cells are columnar epithelium cells that provide mechanical as well as environmental and chemical support for the olfactory receptor neurons. In addition, they contribute to the secretions of the overlying mucus that may play a role in the binding or inactivation of odorant molecules. Finally, basal cells are undifferentiated stem cells that are responsible for the regeneration of both the neuronal and supporting cells. In fact, olfactory receptor cells undergo continuous turnover, with an average life of 1 to 2 months. And they are replaced by new receptors arising from undifferentiated basal cells by mitotic division. The unmyelinated axon of an olfactory receptor neuron pass through the lamina propria and group together into bundles called olfactory phyla, which collectively make up the olfactory nerve, cranial nerve 1. These olfactory phyla pass through the cribriform plate to terminate in the olfactory bulb. The olfactory bulb is a forebrain structure that consists of 5 well-defined layers of cells and fibers, which give it a laminated appearance. These layers and the names of the cells are not required to be memorized. Again, it's not important to memorize the names of the layers or the cells in the olfactory bulb. To continue with the olfactory pathway, the axons of the olfactory receptor neurons synapses with the second-order neurons found in the olfactory bulb. However, these connections happen in a unique manner which has both convergence and divergence. The axons of several receptor neurons will converge on single second-order neuron, which form what called glomeruli, or a glomerulus is single. However, the same type of receptor neuron can also target several glomeruli. This formation will increase the complexity of olfactory sensation by making a combination of different receptor and let us recognize millions of smells. All this happen and processed at the level of olfactory bulb. Also, in the olfactory bulb you can find fibers called centrifugal fibers. These fibers provide feedback to the olfactory bulb from the central nervous system and influence the processing taking place in the olfactory bulb by either sensitization or desensitization, in other words, adaptation. If you put some perfume in the morning, you smelled it for a couple of minutes, then you don't smell it anymore. This is due to these fibers coming from the central nervous system to the olfactory bulb and performs inhibition of the glomeruli at the second-order neurons, stopping it from sending signals, and hence you stop smelling the perfume anymore. Many of these centrifugal fibers are originated from the subcortical division of the central nervous system, hence this process happens in unconscious or involuntary manner. Continuing with the olfaction pathway. The axons of second-order neurons in the olfactory bulb will form the olfactory tract. The axons in the olfactory tract can be divided onto two different parts. First, the medial part, which synapses with what called the anterior olfactory nucleus. This nucleus and its fibers will project to both the ipsilateral and contralateral olfactory bulb and influence the processing happen there. Second, the lateral part, which will project directly to the central areas. Several olfactory projections to the central will target subcortical areas such as olfactory tubercle, piriform cortex, anterior cortical amygdaloid nucleus, periamygdaloid cortex, and lateral entorhinal, later ending up in the hypothalamus. Keep in mind that although these areas has the word cortex in their names, they are unconscious part of the brain, as they are paleocortex, which phylogenetically older type of cortex that has 3 cell layers. These areas will be responsible for the involuntarily aspect of smell sensation, such as emotions associated with certain smells, feeding reflexes and sexual behaviors. Nevertheless, almost 2/3 of the olfactory tract will go to the dorsomedial thalamic nucleus and terminate in the primary olfactory cortex located at inferior and medial surface of temporal lobe. This neocortical connection of olfaction is important for conscious discrimination and identification of odors. Complete the loss of smell sensation is called anosmia, while decreased sensitivity to odorants is called hyposmia, olfactory hypesthesia. The most common cause is upper respiratory tract infections, and sinus disease. Nasal and paranasal diseases such, rhinitis, sinusitis, may block the access of odorants to the olfactory epithelium, leading to decreased olfactory sensation. In addition, edema of the olfactory epithelium may be partly responsible for the olfactory dysfunction. Head trauma can produce olfactory deficits by damaging central olfactory pathways or olfactory receptor axons as they pass through the cribriform plate. Blows to the head can produce shearing movements of the olfactory bulb relative to the cribriform plate. Transection of the thin axons comprising the olfactory phylum may result in a partial or total loss of smell. In many cases, this is temporary condition, as the basal cells will form new receptor and regenerate the pathway, and sensation will regain in couple of months, but in some cases the sensation loss will be permanent condition. Olfactory hyposmia can also be associated with nasal polyps. Nasal polyps are non-cancerous growths that occur in the nasal cavity or within the sinuses. A growth in a sinus may extend into the nasal cavity. Nasal polyps may result from inflamed mucous membranes or allergic reactions and can obstruct the nasal cavities. In these locations, polyps obstruct the flow of air and may cause a number of clinical problems, including a significant compromise of olfactory sense.